All right, team. This is Georgia for the 8 to 12. Saying goodbye. I'm going to go and have some lunch.
Sorry. Oh. I was just saying, when it swap seats when he gets going on the next line. So. Yep, no problem. Oh.
Anything else?
Yeah, we're watching for lounge. Not yet, no. We're mapping right now. Once we're done,
This is a very large map.
yeah. It's been going for a while.
She was resting
All right, we're ready to step over, so go for it.
They're ready to step over.
Oh, yeah.
They're ready to step over.
Suna. Could you tell Suna to
Hey,
grab me?
Scott, do you know that they're ready to step over? No one.
Alexa, drive forward. She is today. I just told Bob we should add that feature. Voice activated.
Insert
At
smart
least for-
speaker name here.
For steps.
Find the Samoan clipper.
All right, so.
Hello, everyone. Welcome to the 12 to 4.
Hello.
Hello.
All right, whenever you're ready. 25, I think. Who's doing?
25. Coming forward.
Perfect. Hopefully we only have 5 more lines, something like that, and then wrap this up.
It's a short dive plan.
I was trying to be concise.
You did good.
A lot of this is active. It's all low. There's all diffuse flow through here. Yeah.
Yeah.
Yeah. Yeah. It's all.
I hope we're above it. We already went over it.
I don't know. They're not that tall. They're only like this, so you might have to like.
Deceiving. Lots of fluid coming out of those. I'm going to guess 70 degrees when we get there. That's my guess.
Are we doing Price is Right rules? Should I go 71?
You would.
No, I think they're 112.
As long as somebody doesn't bet 1 degree.
I'm going to guess. What did you say, Scott? 70?
70.
I'm going to guess like 50.
Ooh, all right.
Lower.
Well, Scott's at 70. Lily's at 71. I'm going with the solid 72.
No, I'm doing. I said 112.
When you think about it, though,
I'm not allowed to guess.
Scott's in charge, so he can really put the temp probe wherever he wants. Disqualified.
Actually, now I'm like,
all right,
50
fair
feels
enough.
kind of low.
I'm going to go bold and say 115.
115?
So
Yeah.
what's our current depth?
The boldest.
All
We
right.
are at 676.
676? Perfect.
How's our tether? Wow, it's murky here. Looks like we're doing pretty good.
And this is still our first photo mosaic, right? Okay.
The photo mosaic.
Oh, okay, yeah.
Yes. I don't think he dropped a point, though.
7 meters to go.
Maybe.
Yeah, Cuskeel.
Let's see how we do with this thing.
So by the time we're done, how big will this whole grid be?
35 by 25, something like that.
Wow.
Wow.
Oh boy.
That's a tall one.
Yeah. Yeah, that's close.
So
3
Chris,
meters to go.
if we have to like change altitude above because we have, some of these are much larger, does that alter or make it more difficult to kind of stitch the images together?
Yeah, a little bit. Yeah, the flat grounds go pretty well. The big spiky bits are harder to deal with.
Last meter.
Nice.
Yeah, looks like we're all right. So yeah, we're doing 1 1/4 steps.
Yeah.
Yeah.
I didn't do it.
Did what? What do you do? Oh, yeah.
All right, step and a half.
Yep, go for it.
I'm loving this purple ship.
On what? Oh, sure.
Eventually. It's the white box on the bottom. Yeah, click that one.
Bob, have you seen this? Oops, yeah.
And then just make it tiny and you know what to do. Did you choose us this purple ship? I like this purple ship. It's excellent.
Summer approved.
We'll give you the credit. It's visually appealing.
So thanks to everyone joining us. This is our second dive on Bailu'u, and this is actually our second dive on this hydrothermal event. This is the last time we discovered it,
So
based on sonar mapping, and we followed the sonar returns on what appeared to
Is
be
there a way to
bubbles,
start it?
yeah, it's on.
and then we were able to get some samples, and then right before we finished our dive, and then now we're back, and we're creating a quite large photo mosaic so we can map this area. That way it can be better understood and documented. And after this, we'll be collecting some samples. You can see some extremophile microbes right there, and those white lines going across the seafloor. Continue to send in those questions or any questions you might have. So what you're seeing, this is only the second time ever this has been viewed. So Chris, these photo mosaics, do they ever become publicly available?
Yeah, this will end up in a publication for sure at some point. And then all the raw images are available to anybody who wants them scientifically through the OET distribution. And then OET will be, if it's a product, can get hosted and people can request the product as well.
Five meters to go. Oh yeah, up underneath those flanges.
Sort of looking for a flange that's high enough and with enough lower topography to come in and look up.
Okay. With some nice shimmering water.
We'll see if we got pools.
Yeah. The mirrors.
We should have reflecting pools.
Yeah, those are cool. Chris, you want to do another one?
Yeah, I think we're gonna go up and back, and then that might be it. We'll see.
Okay. Step into port.
And actually, the only thing we'll do after that, we'll probably we'll turn and we'll run. Let's see, where are we gonna stop? If we go up and back, we're gonna run off to the east to go through the lines we just did as a crossing line.
Okay.
You know what I mean?
Yeah.
And then we'll be out of Dodge.
You want to do the crossing line kind of somewhat in the middle?
Yeah, so we can, that's how. So maybe we'll go up, back, and then like partially up, and then turn right, and then.
Okay. Yeah.
So, the high temp? Got it.
What was the temperature there at that high temp? Okay.
Ooh, I'm in the lead.
Is that that's and
According to me?
that's the that's the one we did
Last
on our
time?
last watch. Yeah, when we were here. 207.4. 207.
I thought it was 202.
202.
202.7.
202.7.
There
Yeah.
you go.
202.7.
Which is roughly 398 degrees
Yeah.
Fahrenheit.
Just dyslexia. Yeah.
Yeah, Hana confirmed 202.7.
That would be intense.
Lily, we have a question coming in about, obviously you know about that first vent that we first came upon before we came over here. About how far apart are those? Because they kind of look like they're opposite sides of
They
the
ditch a plate.
rim of the crater.
Sorry.
Oh, the first event we landed on?
Yeah, the one that
Let's
we landed
see.
on originally on our first dive before coming over here.
Yeah. They are roughly.
So you need an even number of people to make it correct. Yeah.
1,500 meters, so a kilometer and a half.
About a kilometer and a half.
Yeah, and we don't have topography on this. Let me get topography and we can show the viewers. So give me a minute.
What's the name of that one so I can pull it up?
Hype survey. But can you wait till I prepare it?
Yes, I can.
Cool.
Scott, can you pull up on my view the utility for, just like get that out of the way for a bit? Thank you.
Mm-hmm.
And to slurp them.
So Scott, we got to figure out what 202.7 is in Schottenheit.
Ah.
I guess like, wait, Scott, what's
Scotsies?
your
Temperature to
average
length
body
conversion?
temperature? Yeah. Because like, average human body temperature is like what?
98.6.
98.6?
But I'm lower like 98.8.
Yeah, so it's roughly for Scotten Heights. Because it's 98 times 4 is pretty close to 398 degrees. Give or take, like 6 degrees Fahrenheit.
So we're about 4 Schottenheit then, if I hear you.
We definitely need to make an app for this.
Hold on. Let me pull up a calculator.
There's an app for that.
All right, one more step over to port.
Yep, let's do it.
398 divided by 98 is 4.061 Scotten Heights. gotta take it to the third decimal place for precision.
How many Scots?
All right, step and a half, 25.
Yep.
Awesome.
Can I still
How many more lines do we have?
Yeah, go ahead.
We couldn't.
Well, this might be our last one, south. We might do
Awesome.
half of one north, we'll see.
Okay.
7.5.
On that half step forward, do you want me to step over to 1.2 or just continue that? Like stay on the same line?
No, we'll go over to port,
over,
I think.
okay.
We'll just double check. I think we're over the pagodas. There's
yeah.
a few more veiny things out to the left, but we might not
Oh no, that was
chase
the wrong
those all
one.
down.
Yeah.
And then we'll do a crossing line back east. It'll deliver you back to the high temperature vent.
Okay, good deal.
It's a little too intense for me. Let's unload that.
Looks like a fingerprint.
Yeah, sorry, we were talking about the map Lily pulled up, which
There
just
we go.
looked very edgy.
Let's put a colorful one on. That's
Yeah.
the backscatter.
So we have a question about what these vents are ultimately made of.
There we go.
I don't know the exact
Hey.
mineral composition, but so when water is underneath the crust of the earth, it's superheated, so it's able to basically leach minerals out of the surrounding rock.
You put it up?
So what you're seeing is that that mineral content basically depositing into those peaks as the water cools down. I, again, am not an expert, so
it's
if
basically
anyone else
iron oxide. It's like rust.
Yeah.
It's essentially crumbly rust.
And in some areas, there's other minerals as well, right?
Yeah, depending on,
Yeah.
like the super high temperatures have a lot of different.
Yeah,
other-
yeah.
You get actually a lot of rare
viewers-
earths, and you get all kinds of other.
For the viewers, we got the crater topography up on
Thank you, Chris.
3. And so we are to this black blob to the right. And the other crater is over, the other vent was over where these other targets are. So it's approximately 1.5 kilometers between the two.
Thanks for showing that, Lily.
And this is the, yeah, that's the whole crater.
It's also a really pretty map.
It's a cool
That is.
map. Maps are cool.
So all the way to the right
Not
side where
biased.
there's those gaps, is that just a
This?
place that didn't get mapped, or the data was kind of inconsistent?
Yeah, mostly because it didn't fully get mapped. Those are the turning points of the ship. Wait, let me see. Yes.
I believe it is just a gap where the ship
Yeah,
hadn't
so-
passed.
Yeah, it's where the, I just wasn't sure if this was our data or the data we got from Okeanos.
Gotcha.
But, yeah, when the ship turns, the sonar ranges fan out, so you get random spots like that, but you get a lot of gaps.
Got it.
So you want to ensure that you have, like, at least 50% overlap in each ray when you're going back and forth in this lawnmower
Yeah,
pattern.
Scott. So we'll shift over. You can go up to that 3260
mapping.
line.
Hey, Lily.
So
Do you know if the...
say go
Step
12 meters
Oh, wait,
forward
sorry.
and see what happens.
Okay, so I'll step into port, and then we'll go 12 up.
Yeah, and that'll be about halfway, and then we'll
Okay.
see what happens.
Anthony, I've got a question for you that you might like to answer.
Uh-huh.
I don't know if you want to read it.
Okay, here comes 12 forward.
Yeah. And then we'll head east.
Hi, Mom. I know they're not underwater stalagmites, but I'm also not a scientist, so I think I should let a scientist answer what they are. They're what, dormant hydrothermal vents?
Some of them are
Yep, some are still active.
dormant, but some of them
Are
are
they?
still active. Yeah, we saw
Oh, yeah, they're still active.
warm water seeping from some of them.
So we're going to
earlier.
go up.
Yeah, yeah.
We're going to turn right. We're going to go across this survey, and then you'll be back at the high temperature vent.
So, are we going to the high temp point, or are we going to
The main vent.
the main vent? Okay.
Okay. The main event.
But we're doing a crossing line across
my
these
God.
surveys, and then I think so. I mean, not too worried about this stuff again. You could go on for another hour, but I think exactly we're not gonna.
So Lily, question.
Yes.
Do you know if the topography of the Nafanua, am I pronouncing that correctly? The cone?
Sorry,
It's-
I couldn't hear you. What?
I
There
have
we
to
go.
look at the.
The
Ooh, there's a shark in
volcanic
Argus
cone?
view.
Oh,
Oh, there is.
Whoa.
Oh.
Thresher shark.
Oh, wow.
Awesome.
That
That
is a
was
thresher shark.
cool.
Oh, my goodness.
He was
Oh,
there
I gotta
before.
get that.
All right, Scott. So
Did you take a
spin.
shot, Pete?
77
Yes, I
meters
did.
down,
Yes.
there's a
I
shark.
want to grab
So
that later.
Spin heading right.
When you're done,
677
this-
That meters.
Awesome.
Wow.
Oh, thresher sharks are so cool.
That was
They're
super
amazing.
cool. He looked pretty
Sorry,
big too.
I
See that.
All right, heading
Come over and
to
say hi, thresher.
heading to the east.
He's gone.
Yeah. Maybe make your course like 100, something like that, just to go a little bit south.
Sorry,
Okay.
And then
Nafanua
Lily
cone.
and
Yeah, enough.
he's gonna go
enough
east. So you have to pay attention to the ship. He's gonna kind of come
But
under
you-
Argus.
Yep.
Yeah, because like,
So-
oh no, ooh. I'm not the best person to ask about Samoan pronunciations.
But I guess my question is just, do we know if the topography of the cone changed at all between us mapping it recently and then Okeanos mapping it in 2017?
Actually, that dark map that I
Right.
pulled up, the
As soon as I get done doing this line, we'll spin out.
Yeah.
Black
Oh,
and
no,
white
wait.
one.
You can just probably punch in
That was the
30,
back
Scott,
scatter,
and
never
probably
mind.
end it,
Oh.
we'll end it short.
Rennie does have a
But come
difference
around to
map,
maybe 100
Yeah.
degrees, 100.
that I
Okay.
don't think I can load right now. I'm sorry, I need to get the ship moving, but
Yeah,
I
no
will
worries.
answer.
Rennie had showed it to me and it didn't look to be
Sure.
like there was any
Is that the end
areas of
of
major
the-
differences.
All right, yeah, just punch in 30 and go.
Okay,
Okay.
here
Is
comes
this
30.
end?
No, this is our crossing line, one
Okay.
line back across everything.
And at the end of this, I need to spin a wrap out.
Can you put, once
Oh,
we're
the cover.
done, put the final dimension of the plot on the
So
note?
which way are you facing?
I'm
On
facing.
here? Yes.
Cool. Thank you.
Yeah, of course.
Yeah. Okay.
Brian, do we have a lot of viewers tapping in for this dive?
We're about 147 right now.
Oh, wow.
Awesome.
Come on, 12 to few viewers.
Join us. It's our last one
Bring
of
those
the
questions. This is it.
Last one
Yeah.
of this cruise.
Thank you for joining us.
US, UK, Canada, Australia, Netherlands, Japan, Russia, Denmark, Portugal, New Zealand, Norway, and Germany. We're looking at you.
Wow.
Woo!
Thank you, guys.
I
do.
am so badly hoping that Thresher Shark comes into view
I
again
really
too.
hope it
Yeah,
does.
you got a couple grabs though, right?
I got one,
Time.
yeah.
It
It's
was
a little hard to make out because of the
23.
just like
Yeah.
turbidity in the water.
It was
but,
kind of
like,
dark too. It was on the lower
super
corner.
cool.
Thresher Shark and
DC
hydrothermal
graphics are
vents,
looking awesome.
1-2
Yeah.
punch, that's hard to beat.
I don't-
Yeah.
wanna-
I have some
forgot to ask
Thresher
what they
Shark
did.
facts, but I want to
They-
fact check them before I show them.
Right. Yep. Yep.
Peyton, is Rebecca our Extremophile super fan back there still
She
with you?
is.
She's here.
She's supervising.
Rebecca, is there anything that you'd like to share with our audience about Extremophiles?
Rebecca, is there anything
Quick.
that you'd like to
Super
share with
quick.
our audience about Extremophiles?
Especially ones around the hydrothermal vents.
Yeah. Yep.
All right, she's hopping in. Hold on, one second.
Nice. Rebecca, you can't expect us to be back there and not join the conversation. You're
Hello.
You're an honorary
Hi.
Benthic buddy now. So is there anything that you'd like to share with the audience about some of the extremophiles they might be seeing here at this hydrothermal vent?
Sure, yeah. So right now it looks like we're looking at some white and black bacterial mats. The brown ones are probably also a different type of microbial eating the iron oxide, the brown deposits there. The white ones are most likely sulfur-eating bacteria. A common one is Bagiatoa.
Okay.
It's, I mean, you can't really identify them with a video camera because they're so small. But they are pretty large bacteria. They form filaments and they're actually clear. But when the sulfur compounds build up inside them, that's when you get the white color.
Interesting.
So that's what we're seeing here.
That just answered my next question. Good job, Buffler. So what got you so excited about microbes and extremophiles?
I've always been interested in the deep ocean from some of the Blue Planet documentaries, and my favorite episode was the deep
You want to
ocean,
flush
which
it now?
included the hydrothermal vents.
Just to be ready.
And I think I've
Yeah,
been
let's
hooked on
get
it
it.
ever since.
Let's get it ready.
Now, why is it so important for humans to understand more about these organisms that live
Oh,
in
that's
such
a
extreme
great
environments?
question. Yeah, so because they live in extreme environments compared to like a human level, they can do a lot of cool stuff that you can utilize in biotechnology. Their proteins and enzymes can function at a higher temperature, which
Yeah.
chemical reactions
Maybe
generally
maybe
work
extend it
faster at
another
a higher
5.
temperature.
What happens to normal proteins when they reach higher temperatures?
They denature or basically unfold themselves and they can't function anymore. So it's really cool to take different properties from these microbes and then utilize them in biotechnology.
So,
So that's a very common property of them.
can you give an example of maybe like how something from an extremophile like this was used in biotech?
Yeah, so one of the more common examples is a DNA polymerase from an extremophile that lives in hot springs. It's called TAC polymerase, and it's from a thermophile, and it was isolated from a microbe from Yellowstone National
Yeah, maybe
Park.
another 5 after
So
that.
this is used to amplify small amounts of DNA into large quantities of DNA, which is very useful for a lot of experimental techniques.
Nice.
And that's a component of like PCR, right?
Yeah, PCR or polymerase chain reaction, which is a common, again, another common laboratory technique.
And what did they use PCR for?
This is the one that you can amplify small amounts of DNA into larger amounts to the point where you can visualize it and see it with your naked eye.
So CSI type things or
To see what?
like crime scene investigation type things?
Yep. So if you have a small amount of DNA, you can amplify it and then do further studies to compare it to different people.
Sure.
Let's see. My favorite part about extremophiles, though, is the astrobiology applications. So there's a lot of environments off Earth that more closely mimic kind of these conditions: high pressure, high temperature at the vents, low temperature outside of the vents, with one example being Europa, moon of Jupiter.
Did we end the survey?
And that was 5. Sorry.
Okay. I'm sorry to interrupt.
Oh, no, you're good. You're good.
Yeah.
I'm not talking. Go ahead, 5 more. Yeah, let's see here. Just to make sure we really got it. And then
So by exploring
Quite the
the
survey.
environments and microbes down here and designing technology to investigate them, learn how to detect them, learn what to detect, we can then use that off-Earth missions to search for life elsewhere.
yeah, no, exactly. It's,
Awesome.
I
Thanks,
think
Rebecca.
the main vent's like around here. But
It's a solid
Maybe come down.
20 meters behind us now. Whoa, we just jumped. Not. Yeah, it's like 30 meters behind us.
all right, so when this settles out, we're good.
All right, I'm going to give the mic back to Hannah.
All right, that's good. So yeah,
See you,
I'll shut the
Rebecca.
cameras down. You guys are good. If you spin around and head back, I think a little bit right, you should be right around the main
Okay.
vent.
Now is this end?
275.
All right, turn into port.
Going to port? Okay.
Gotta stop.
270. Just.
I'm sorry, I keep
Directly
asking this,
west.
but is this end of transect?
Yes.
Sorry.
Okay, thank you. End time 2323. Depth is 675.8. Right. I'm going to say total was about 20 by 25 meters.
You could say, yeah, 25 by 30 is fine.
25 by
Plus or
30.
minus.
So about
There's
12 and a
roughly.
half by 15 Scott. That's a lot of square Scott.
It's
a lot of square
area. Okay.
3 meters high, so 1.5 Scott, so we get a nice Scott cubed.
Testing,
So-
testing. Do you hear me?
Hello.
Loud and
Okay,
clear.
super. Thank you.
So we are currently returning to the main hydrothermal vent to collect biological samples. Is that correct?
Yep.
As well as deposits.
Yes. Thank you.
What's on the top of our list to pick up, Peyton?
We have a number of things. One thing we want to do is collect a Niskin bottle right above the high temperature vent. So
Okay.
a Niskin bottle is basically a cylinder that has caps on either side that can close. So we're able to basically get samples of seawater at any given depth. We're also looking to collect push cores of different microbial mats. There's also some piece of like rusty sulfite, so maybe some of the material off the towers that we're seeing or in the rocks.
Yeah, we
themselves.
also want to collect the macrofauna.
Yes, so we're looking
there's
for
shrimp,
the
2 species of, maybe multiple species of shrimp. Certainly 2 apparently different crabs. There's a white one and a brown one.
Do something to stop the cameras.
There are
Chris is controlling them.
Oh,
Are they
okay.
flashing?
No.
We saw
never mind.
isopods.
Yeah.
There's pink isopods.
I just
and then we'll see what else we see.
autopilot.
Yeah.
Have all the species that we've seen already been identified?
We've only collected 1 crab, and I don't think it has been identified. The shrimp will be the tough ones. The isopods will be pretty easy. We'd like to get 3 of each.
Yes. I'm actually really excited about seeing these shrimp in the wet lab because based on what I saw last time.
Wait, you're excited about the shrimp?
No way. Yeah, I'm excited about the shrimp. But these shrimp in particular were really interesting because they didn't have visible eyes. And other species in the same genus, I tentatively identified it as Remicaris, but they have a pigment that lets them sense what we think is black body radiation, so they're able to sense like heat from hydrothermal vents.
Cindy Van Dover, Duke.
Super cool.
Yes, Cindy Van Dover, by the way, was first female Alvin pilot before she got her PhD.
Was she the person that described it?
Yes.
Genus? Okay, gotcha. But very exciting.
From the Cayman Trough, which we first explored in 1976. The leaky transform fault.
Okay, it's gonna be, I'm going to this one.
Viewers continue to send
Are
in
we
those
done,
questions.
Chris? Okay. Should see
It
a
is
little
10
mound.
meters to 263.
263?
Yeah, and it was off by 7 meters, so you're close.
Oh, it was?
Yeah. Well, unless this is a drop, as opposed to the original anomaly.
What's that sonar
If it's
target?
a drop target, then it should be
I
dead
don't know,
on.
that's what I was wondering.
It should be dead on if it's a drop target when we first came in.
It's to the right?
It's a little volcano, or little mound.
Are you going in?
Yeah, keep going. You'll keep going in on that drop target.
So you have the target behind me? Or
it's
263?
Let's do 215.
215?
215, it's 10 meters.
Okay.
Yeah, I don't know what that target is.
I think it is off to the right.
Yeah, I see it.
There it is.
Got the visual.
Okay, now again, the shimmering will affect you on, so you have to go in and see what way it's being pushed.
Do we have a preference for what order we want to collect the samples in?
Use it way in on
As they
FG.
present themselves.
Okay.
So well, basically, I think if you go down and land here, facing the way you're facing, that seems to have the most diversity when we last looked at it. So I'd go straight down and come up to it, and then land.
All right, current's still going south.
Yeah, let's just see if the shimmer affects you, but I think it's going, it won't
All right, you
affect
can
you.
zoom back out.
So viewers, what you're seeing is the hydrothermal event. This is the second time ever anyone's ever seen it.
So you'll
Previous
see that
times-
little embayment there in the mound, Herc.
Okay.
See
Cool.
that little cul-de-sac up there? The little embayment in the vent is just to the right of that rock. You'll see a little spot. You'll see the isopod there, the pink one. Just go in. It's just, yeah, I would park myself within reach of all of that.
It looks like some of the structure
Maybe we
collapsed
could,
since last time we were here.
Yeah.
I'm gonna
Grab
get a-
some of the
Where's the
crabs and
pointer?
then slurp the isopods and the
What
shrimp.
happened to the pointer?
I don't know what, Scott, what you think?
Do you know what happened to the pointer?
No,
The
Stick
stick
of Science was over there.
It
Should
vanished.
be there somewhere.
It's not
I
that
need
back
some more
corner.
leash, actually. Yeah.
It does look like the top part of that vent collapsed.
Yeah.
It definitely does.
Bridge nav, can we
It
have
was-
a
I want to use the broom, but I guess I'll use the broom. We have a pointer.
285?
We had
It was
one.
in the corner over there.
Yeah, I don't know where it
Oh
went.
well, I'll use the broom.
We got the stick of science broom.
Yeah, you want to zoom in on that. I think it's a crab.
I'm getting
Wonder what-
thwacked.
Not yet. Only if you misbehave.
So here, as you can see, Dr. Ballard using the broom of science to help us
Wow.
Zoom
identify.
in half on Argus. You go with that?
Yeah, I'm good.
Do you want to zoom in half on, or
Yes.
as much
Where
as
the
it looks
heck is
good?
that crab pointing?
So we
living
want to
over
get aim for 3 of each. So 3 of the white crabs,
That's
3
a
of
cool
the
Argus
orange
shot.
brown juice color crabs,
Okay.
Very cool Argus shot.
and then the isopods and the shrimp.
Yeah.
The shrimp will probably
Just
be the
heaved.
hardest ones to snag.
Not for Zeus.
And
Not
zoom
for
in
Zeus.
on Herc. Okay. Well, I see
See those isopods right there at
Oh,
the
there
bottom?
they
we got a twofer right there.
Yeah,
Oh,
two
there's a bunch of shrimp right at the bottom too.
Oh,
Yeah.
maybe you could. Oh, there's 3 shrimp right there.
Yeah.
Maybe you could 1, 2, 3, and then the 2 isopods.
All right.
And
Excuse me.
I shall do my best.
All right, we're in the big mesh, chamber 7.
And zoom wide.
Big mesh? Okay, thank you very much.
I don't think it'll melt your.
Let's hope not. Oh, the 2 crabs are fighting.
Time to break it up
Time to break it up.
with the slurp tube.
So when we took a temperature reading the other day, the temperature was 202
Okay, ready to slurp?
degrees C
Yep.
7.
at the top of the vent.
Slurp is on.
And go for zoom.
And the isopods will be pretty easy.
Oh, the shrimp.
Yeah.
They're on to us.
Yeah, but sometimes they go in a hole and you stick it in and get them. Oops.
Off.
You don't want to jam it. Is the pump on?
Yeah.
Off.
Maybe.
Off.
Oh, sorry.
Yeah, it's stuck. There you
Okay, on.
It's on again.
go. Yeah, it's churning. Those guys right there. We got 2 of them.
There's
Got
a one.
2.
Isopod.
Now there's a
2
to the
isopods.
shrimp. To the shrimp, just under. They're right there. Get them. You got
Oh.
one.
Oh, you got
Oh, nice.
I think, was that 2 or
Yes,
1?
I think that
two
was
it looked
2.
like.
Okay.
If you go underneath, your
And
sucker,
you can grab the crab.
there's
over there.
another shrimp right there on the rock.
To the right there.
Yeah.
Yep.
There you
I
go.
think you got him.
Off.
Oh.
Just temporarily.
Off,
And then on.
on.
Okay, so that's 3 shrimp,
What do you
3
think?
shrimp, 2 isopods.
0 crabs. Anybody
The
see any
smaller
more
crabs are good. You
isopods?
got 1. We
What
don't
color was
care.
that?
There's that one.
That's a
little
white
one.
crab.
Are good.
Oh,
Got
Oh,
2.
2
Two.
crabs.
so that's 3 white crabs.
There's a shrimp.
No, one was brown.
So are the brown crab and the white crabs different species or just colors?
We're
Find
not
out.
sure.
Certain.
That's why we're sampling them both.
Find out.
You okay over there?
Was
To be
above 40.
determined.
Oh.
Okay, I would go err on getting more because you know we're not gonna know. I can sort of see them in the bottom of
There's a small white one. Okay, so that's 3 white I have for count.
Okay, did we just collect another white one?
We just
Yes.
did right
We're
now.
Okay,
erring
that's-
on
I
the
think that's
side
4.
of
Oh,
getting more than 3 if we won't.
Oh.
I've sampled them and have them. We sampled an octopod
Nice.
and he figured his way out. Okay, let's reconnoiter here for a shrimp.
I don't know if I can reach, but there's a scale worm over there. Or
Yeah, I would go for that.
oh yeah.
You want to zoom in on it?
I can't reach.
The little guys.
Let me try again.
That pink thing?
Yeah.
I think that's an isopod.
Alright, that's 3.
And you said you
So-
wanted some brown?
Yeah.
Yeah. So I got 3 isopods, 3 shrimp, 4 white crabs, and 1 brown crab we have so far.
One, there's
Okay.
a brown.
2 brown. 3 brown.
Go for another one. There you go.
4
4
brown.
brown.
Okay, so,
So
let's
let's-
look for shrimp.
Another shrimp, yeah.
You might want to zoom back and look around with your camera while we're sitting here.
Yeah.
Let's
Maybe
see.
to the left.
That's a very
It's hard to
active
see with all the
search
shimmer
right
coming
there.
our way.
Yeah.
Hopefully the crabs don't scavenge
Zoom in
any
just
of the
like
other
half zoom.
animals.
What are the chances they eat each other?
Yeah,
They
that's what
would
I
have
was
done
thinking
it
a
while
little
they
bit.
were living here.
This is true.
They're probably sort of discussing an escape strategy anyway. Okay, let's see. I don't see. The shrimp tend to get smart and go on the rocks. Keep looking. They tend to be more peripheral too. You might bump a rock or two and
I don't see any.
shoot one out.
Huh. Oh, there's another isopod dead
Yeah.
center of
Yeah.
the screen,
Dead
12
center
o'clock.
by the-
Got
yeah,
him. Suction on.
yeah.
It's on.
Four iso.
Oh,
Oh, in escape.
Oh, come on.
Oh,
Got it.
There we go.
That's up to 4
Nothing
isopods.
personal.
correct. That's the fourth one, I
Crafty.
believe.
Okay.
Nothing
Here,
personal.
shrimpy,
Shrimp,
shrimpies.
Come to Papa. Come on up. Yeah, just bump the rocks a little.
Can we shut suction off?
Yeah.
They
So
tend
we
to-
don't
Oh,
suck up
yeah,
any rocks and clog everything.
Why don't you, while we're in this mode, go around to the other side.
Yeah.
Can I get a zoom out on Argus? Should I take a step?
Active
Do we
vent.
need more tether to get to the other side?
I'm pretty
You could
much
maybe
over
name it
him because I'm looking
northeast.
straight down.
Oh, you are?
I think
Okay.
Yeah.
I think
North-
we're
Is that
good.
another
I think
crab?
that's the location of it,
Nav
northeast
G2.
vent.
Can I do a reset?
Yes. Yes, you can.
Oh, there was an isopod.
I was just- I think I
See
just-
that that
Bottom
by
left
that, left by that.
by the
want-
white
right
crab.
You want that one?
Yeah.
Okay.
It's a big guy. So that'll be 5 isopods.
Maybe 2
Okay.
there. If you
Is
want
there
to
a
zoom
second
and you
one?
can see
There's a shrimp, maybe?
Where?
one.
I can't tell. It was floating kind of at our...
Hey, Peyton, did you number those all differently?
No, those are all one sample, and it's still going. We're still at 107.
107?
Did we
Okay,
just
ready
open
for
up a
suction?
seat
A,
where
B,
we
and
landed?
C, D.
What?
I don't know, maybe it looks like it.
Suction
some
on.
bubbles came.
And zoom.
Ooh, that's a cool thing.
Goodbye. All right, that
It's a
is
big guy.
5 isopods.
You'll live
Alright, now
today,
let's go
crab.
around the corner, see if we can spot more shrimp.
What if you accidentally sucked it in right as you
Suction
said
off.
Thank you. Okay, go around the corner.
Yeah, and then we're going to want to take a couple samples of the deposits.
For a viewer that
Different
is
colors.
Dr. Ballard, you hear?
Yeah, do you hear me?
It's air bubble. Oh,
Viewers
yeah.
are quite
We're
excited.
at a depth where they can gas can form.
Yeah, it was stuck on bubble cam, and then it just went away.
There was a bubble on
Yeah.
bubble cam? Is that irony?
It was
just
very
blew my
You should see
mind.
when we look at that. You should see when we completely bury the brow in
I don't know if I want to see
hydrates.
that.
It looks like,
Is that a good thing?
What is that right
Ice.
there? By
Yeah,
underneath
this-
the left laser.
Oh, I see it.
Is that a worm?
I can't tell.
I
I think your best bet would be
Don't-
up in that.
don't
You wanna go up
know.
there?
Yeah.
Doesn't.
Oh, there's a
stop-
shrimp.
They tend to
Right
be
by the
where
drop
it's
plate.
high temperature.
Where is he?
It was right
let's
underneath the drop
just-
plate we were
Oh,
Zoom in a
missed
little
it.
because they're hard to see.
Yeah, have zoom.
they are on to us.
There's another
Words
isopod.
out. I think, so all we need is a couple more shrimp, and then we'll shift to rock samples.
I think if I told my friends back home that I was helping to sample deep sea shrimp, they would be like, yeah, that sounds exactly like you.
Oh, there's one.
Oh, there's
Oh,
one
where?
center
by
of
the
the
vent.
screen.
There's
There's
two.
two
Oh,
of
it's
them.
escaping.
One's going to the right. I
yeah,
lost.
see him? He just, see him, yeah, see him over there?
Oh,
Not that guy, just above
Yeah.
him.
I'm all
they're
Might
taunting
wanna
us.
zoom.
jacked up on my arm.
You want to put
There's one
suction
in the
on?
water
Yeah,
column.
suction off.
Suction's on.
Yeah, he's too close. My,
So
arm,
there
my arm
was-
doesn't bend that way.
There's one in the middle of the screen at about 6 o'clock, straight down from where.
He's
You see
under.
him?
If you follow that white crab south,
Zoom.
it's right about there.
2 zoom.
Oh,
Just below where that
There,
one
you're
is.
right there. That one, you're a little guy.
And then
there.
there's one.
And
just
there's
below
one-
you,
Ah.
too.
Come here.
We just cooked him, he can have cooked shrimp.
there's another one right below.
Right.
Yeah.
Right below 6 o'clock.
There you go.
There we go.
Oh, I think we got
I
him.
think you got him.
Yeah, we sort of closed the door behind him. Oh, there's one. See him right.
I'm going to
Okay,
say
so-
4 shrimp.
Yeah.
Right straight down. He just moved, right? You're pointed at him. A little higher up. You're blocking him out, but he's just,
Man,
Wow,
this
I do
slurp.
not see that zoom in.
yeah. You lift up your hand for a second.
There's one
See
to
him
the
on
right.
the rock?
Over there.
Oh, there he is.
Ooh, look
Oh,
at
we're
that.
Nice.
That's 5 shrimp.
Did we need more
Correct.
of
Oh, we
the
never know.
isopods?
Um,
I think
we
we're
have
good.
5 isopods, 4
Okay.
white crabs, 4 brown crabs, 5 shrimp.
I think we're good.
Okay.
So
All right.
let's stash
All right,
that.
section
Do we
off.
want to do a Niskin on top of the plume or wait for
Well, I
that?
think while we're here, we'll grab some rocks, and then on the way out, we
Gotcha.
can do the Niskin.
Sounds like a plan.
Okay, so what we want to do is go for colors. The black one in the middle, that's that guy right there. That's
Marking end of that sample. That was sample 107.
so. Yeah, that's perfect. Whatever you can get. It might be too friable, but
All right, start time 23:42.
it will. Yeah. So you're gonna have to really under grip it. I would start with
We have
that
to scoop
big
on it.
guy where the lasers are, because it's going to be become a small rock. I wouldn't worry about it being a big one right now. It won't remain that way.
What type of temperatures can the arm withstand?
A lot.
That, if you can, yeah, it's going to slowly become a
A
smaller
lot of
rock.
it's titanium.
There you go, perfect.
Okay.
Get the tray
Forward bio
out.
boxes are open.
Don't do much.
Forward.
Because
Yeah, let me get a
it's
couple
going to
grabs on
probably
it.
shatter.
Yeah. Roger that.
Okay.
Ready?
He wants to get grabs of it.
Yeah.
If it's
geez.
going to break, don't worry about
Yeah,
it.
this
I-
is fragile.
I was gonna.
Can I
okay.
get one with the
Sorry.
lasers on it, and
Go,
that'll be
go
it.
for it.
Got it.
Go.
Good.
All right.
Thank you, guys.
Panding down.
Oh, quick, get
sorry,
the door
coming
out.
coming
It's
out.
about to.
Coming
Oh,
out, coming
oh.
out, coming out, coming out.
Yeah, it'll become a bunch of little ones in a second. Right about now. Okay.
Oh, it kind of
Okay.
stayed intact.
Gray crumbly
Okay.
rock pulled directly from hydrothermal vent. Some
Now
bacterial mats
we're gonna
attached.
go for the orange ones.
That was sample 108 and
So I would, the best ones are off to the right. Or let's see what you got. Yeah, someone over there on the outer flanks, the oxidized ones. They're just oxidized from the other guy, but they should be a little harder.
Thank you.
Oh man.
Not a lot. Yeah, they're going to be pretty brittle.
Oh,
What box are we gonna put these in, Peyton?
I put them in a little one.
Nice.
if you guys want to put them in starboard, we can definitely do that.
Starboard.
Okay. Yeah. Starboard bio boxes are
Take
all
a
open.
grab.
Yep, got it. With the lasers, thank you very much.
See if it
Um,
see if it makes it.
rock from edge of vent site. Brown material on gray.
Stand by.
rock.
Camera change.
Ready, camera change.
Okay, and go.
Probably the back ones would fit. What do you think? I think it'll.
Eh.
I think it might
Well,
break up.
maybe the
Close.
front one.
Yeah.
Play it
That
safe.
looks a little big.
There you go, perfect.
Thank you.
So what we want to do is get water. And I've never done this with an Anson bottle. So I don't know if you're going to fry anything on the side of the vehicle.
Should we see what the temperature is above
You could
that?
get above it, but that's going to be pretty difficult.
So we did a sample last dive when we first got this where I was several meters up and just put the bottle in the plume.
So you did get that?
Yeah.
So we got that sample process?
You can do
We,
it,
it was fil-
It
Go
was
ahead,
filtered
Dave.
for eDNA, which I don't think is what we're looking for. So we can grab a Niskin.
Well, you're certainly not going to capture the gas.
Right. Yeah. I guess my question would also be, have we heard back on how they want the water stored once it's collected?
Well, first you collect it.
Okay.
It's on your port side, right?
Yeah.
So we can look and bathe yourself. Yeah.
This is one.
And then see which one's getting it. You might want to get your manipulator ready. Because it looks like it could be
Hard to tell.
Yeah, they're all
interesting.
open, so.
I think it's in the first one.
Yeah, we want to
Closest
get it
us.
ready and not knowing which one it's going to do, but get it over there. Okay, now we'll see which one it comes to. All right, you got 1, 2, 3, 4. Is there 5 of them?
Yes, all of,
there's
or
5.
there's 6 of them. 6 Niskins.
Yeah, there's 5 of them.
Huh?
8, 9, 10,
Oh, but he's
11,
up.
and 12.
We're missing 7. It's not out of view. It's not in view.
There's 7, 8,
There's
9, 10, 11.
Yes, there's 6 Niskins in total.
12.
They're all
Okay, there we go.
available.
All
All
of them are
right.
currently open.
So let's see which one starts bubbling stuff out the top.
It's going to be.
And their 12 is the closest to us.
Yes.
Yes.
Yes. All righty.
How many meters are we above the surface?
2.4.
Yeah.
2.5.
From where the sensor is.
The Doppler. Yeah.
Which is it on the back?
On the back.
Right now it says 2.5.
You guys have 2 data loggers back here trying to record this all physically and electronically. Go,
You
data
can zoom in on
loggers.
Argus.
Where are you getting the Doppler? Oh, gotcha.
You can also record it, yeah. That's the altitude from...
How's that,
Kurt.
Scott?
Good. Thank you.
I'm just going to pull it up here.
I remember when we found the first black smoker and we went in with Alvin, and little did we realize another one was about to fry the downward looking porthole.
Uh-oh.
And it melted the syntactic foam.
Oh, wow.
So here we are.
To be
As
determined.
Yogi Berra would say, deja vu all over again.
I didn't see very many bubbles coming up.
Yeah.
Yeah.
I don't even know if you got over it.
We were at one point. I saw them on the porch, but
All right, I'm going to back up and get a visual on it again.
Yeah. Your iris is pretty wide open on
Yeah,
Herc.
he's got me
So
so I can
it's-
see the
Yeah.
pull strings.
But you can't see the bottom well.
Yeah. All right.
It's to the left, bearing like, I don't know, 290, I'd say.
You're full wide on Argus. So
That's a
Thanks.
chimney. It's off to the right. It's over to your right. There it
There you
is,
go.
dead
There we
ahead.
go.
So you want to graze by it. Making a bombing run.
We're a little closer to 3 meters above it right now. But wait
Yeah,
until we
sorry.
actually sample it.
Yeah.
Furo, thanks for your question. This isn't the last dive of the season, but it is the last dive of this leg.
We have many more dives to go. Should see it in bubble cam, I think. Off to the side anyway. There it is.
Yeah. So there's your front porch just on the left.
All right, so that's right on the corner there. So this should step me forward.
Yeah, I mean, I see bubbles in the Niskin cam now. I don't know if they're going through them, but.
Right.
Well, the flow is.
Yeah.
It looks like there's some small ones coming out of it.
Yeah.
Number 12.
Yeah, I think so too.
Should we pull 12?
What do you guys think?
I don't think so.
Okay.
Just get ready though. I would try to get the flow through your grating up on the nose,
Okay.
so you actually see the bubbles, and just walk it. So I'd try to go through the porch. There, I can see it in the grating. You're over it. Now if you move forward a little, it should go through your.
Oh yeah, there it is.
You can see the reflection. There it is. See it on the porch?
Yeah, down there.
It's right in there.
Yeah.
So then you walk down a little, and it should get to that.
Our altitude's 2.8 meters, 2.7
Yeah,
meters.
it's going. We saw it on a multi-beam going tens of meters.
There's some bubbles through there.
Yeah, I would pop. Let's see, is it 10 or 11?
12. 12 is
12?
the closest one.
But which do you think's getting the bubbles? You're in it.
Maybe 8.
Or
it's hard to tell.
yeah, it's a pretty. I would
Pop 'em all.
no, I would
I'm just
pop.
kidding.
I would pop 10.
You won't put that port camera on your left monitor instead of Argus and put Argus in center?
Sure.
Yeah, that'd be
Yeah, stand
a good
by.
idea.
Can you zoom in with Argus? Real tight?
We were doing
No, all the way in with Argus?
I'm on left RV.
That's your real eyeball in the sky. I think this is a pig and a poke, so go ahead.
All right. Here comes 10.
There we
10
go.
got. It is.
Oh, that was 11.
2354.
Well, that's all
Was that
right.
11?
That's weird.
Yeah, it ended up being 11.
But I pulled.
No,
the-
it was 11, but it's okay.
Oh, look, there's bubbles. Yeah, all coming up behind.
Okay,
All
our
of
number
them.
was 2.5
So
meters
take
above
note.
the
I
fan.
pulled 10, but 11 triggered.
Oh, that's interesting.
Okay.
All right.
Oh, well.
Here's what we're going to do next. We're going to do push cores.
It's in 11.
And we're going to go out on the flats. And we're going to do them in the different colored sediments.
Different color sediments.
Yeah, so let's just get away from this guy and go out onto the flats there. Yep. And you'll see the different colors. White, black, and orange.
Next sample will be 111.
Tangled up in my wires.
I could really use a masseuse right now. Oh, nice few kinks in my back.
All right, push cores at different
Colors.
different colors right up
I
here.
think they're layered. I think the black is the underpinning with white on it and yellow on the white.
Okay, so when I go into the
Yeah,
black here.
yeah. I think you're going to get, if you go orange, you'll get all three, but let's
Okay.
do black first, then white, then orange.
Okay.
We'll see if they stick. They may not stick.
Yeah.
It may be like the other ones where it's
We
a
just had
fine
to scoop
grain
it.
sand, not a mud. Yeah, super.
Nice.
So, there we are.
I'm poking the box out a little bit.
Roger that.
Oh, can we get bubble back on